Okay, so we're going to cover these other words that are part of the jargon of synthetic biology. So one of them, and this is one that has been popping up throughout the whole series of talks, is standards and standardization. So we'll spend some time discussing standardization, and also we will spend some time discussing standardized parts and how you go from standardized parts to form devices and then eventually systems, okay? So just to refresh your memory from the first day, we discussed the first day, or I mentioned the first day, that standards is standardization is one of the key pillars of contemporary engineering, and therefore any attempt to any attempt to make biology amenable to engineering has to go through the process of standardization of some of the tools and some of the methods and some of the things that I will mention along today. Okay, so what a standard means, and I'm going to spend some time discussing with you what is a standard. So a standard originally means a banner, like you go with a banner and then the others follow you, and that's a standard. So in a battle, everyone, or in the past at least, everyone will look at the banner, whether it's there, whether it's not there, and that will orientate the different actions. So that's why the term standards comes from the military world, but then also the meaning of it is all standing for the same. And therefore, keep in mind that the notion of a standard also embodies a degree of leadership, okay? So if I propose a standard and everyone follows me, that means that I am in a position of leadership. If the French Revolution proposed the metro as the standard for a distance, then that means that, you know, they had a kind of leadership in the field. And so keep in mind these two aspects. One of them is a standard is something that everyone recognizes as significant, and also a standard means that the one that holds the banner has a degree of leadership. So what is a standard in engineering? So there are some features of standards. First of all, a standard is about the words, semantic and graphic language for annotating and the nature and the properties of a system. Okay, so when we use logic gates and we use the triangles or the little bubbles or whatever, that's a standard. Then it's also about definition of units of measurement. So you say a degree or 5 degrees or 1 meter or 5 meter or 1 ohm or 1 ampere. This is a standard, okay? In many cases, as you know, not every country has the same standards. For instance, the British, for many years, had their own British system of standards that, by the way, was quite messy and to this day causes some problems. But anyway, so standards is a kind of, I wouldn't say artificial, but it's a kind of choice that a community takes for doing things together. Then another feature of a standard is about geometric shapes. And this is, for instance, this screw turn that I just mentioned before. And, well, this is also a standard, geometric shapes. And also the implementation of protocols. So you prepare something according to a standard. If that standard is well established, the same thing can be repeated here or somewhere else, okay? So this is in general what a standard is about. So what are standards good for? And again, we are still in the realm of engineering. Well, a standard, first of all, enables the abstraction of the properties of the component of a system. If we say I have 5 amperes, you know, you don't talk about the electrons, you don't think about the kind of material that the electrons go back, you just have that idea of amperes. They also enable a precise description of something in a standardized quantitative language. If I say the measure here is, I don't know, 70 centimeters, you know exactly what I'm talking about. And also, because of this graphical aspect of standards, you can represent the composition of an object with identical representation methods, okay? So if I make a logic circuit and I send it to China or to Singapore, it doesn't matter, they will understand exactly what is the content of that particular blueprint. And as I also mentioned on passing the first day, a big bonus of standards is that you can disengage the design of a product from the fabrication, from the final assembly for the distribution. So this is something that is inherent to the value of standards. So what things can be standardized? Okay, there are some things that can be standardized in engineering, and as you will see immediately, some of these things that can be standardized have an instant kind of projection in the biological world. So for instance, you can standardize the physical assembly of the components of a system, and then, for instance, about geometrical shapes, dimensions, boundaries between elements, compositional rules. This is something that can be standardized. Also, you can standardize the metrology, the way you measure things. You use a given technology to measure relevant properties, to calculate units, to give reference values, to examine tolerance and other ones. This is something important that I will return to in a moment. It allows you also to measure or to consider context sensitivity, transfer functions. Okay. You can also standardize the manufacture of engineered objects. And there is an international organization called ISO, that is the International Standards Organization. That is the international body that will give a stamp so that industries can follow given procedures to produce given things. So you say, well, this object has been produced following a standard ISO 300-475. Then that is a very established procedure that can then be reproduced by many others. You can also standardize formal languages. And this is something that in biology, as we know, sometimes is very important. Workflows, data exchange, operative systems, databases. We can also standardize risk assessment considerations, like safety criteria, security benchmarks. We can even standardize ethical appraisal of things. For instance, agreeing on some consensus rule. Can we release into the environment this or that? Can we use RNA vaccines? I mean, these are standards. They are not as physical as a standard for metrology or a standard for electricity, but still they are standards. And also we can standardize the way we handle intellectual property. And as you know, different treaties deal with knowledge protection through patents and open access and all these type of different agreements and so on. So the essence of this is that you have a bunch of possibilities to apply standards to. If you apply them, then things can go much smoother, but there must be an agreement in the community to do that. And this agreement sometimes emerges from top down, so it is the community that agrees to follow a given rule or a given, say, standard, but sometimes is imposed by brute force, okay? And the typical example is the way the metric system was mentioned that on passing the other day. So the reason why we have the metric system in Europe, in continental Europe, Napoleon conquered most of the European by brute force that all countries had to follow the metric system. Before that, every country had a different system. Spain had one, Austria had one, Germany had one. So he said, well, this is a mess. So from now on, by brute force, either you take this standard or you might be in trouble, okay? So and this tension between standards that go from the community to the way up, and also standards that are imposed by brute force is something that happens also in some cases in synthetic biology, as you will see later. I showed you this slide before. This is an early example of civil engineering. It's an aqueduct that is in a city close to Madrid in my country, in Spain, and you can see because every block here follows of size, of angles, of say periodicity, and so on and so on. And thanks to that, it was possible by the Roman architect to design the thing, to produce the stone somewhere else, to assemble them somewhere else, and then deploy the things somewhere else as well. Okay, so as a consequence of that, and this is something also that I mentioned on passing the first day, you have to reshape the way you look at biological systems from the perspective of synthetic biology. So in biological systems, the kind of frame that enables us to understand what is going on in live, objects is the so-called the central dogma of molecular biology, where you have DNA, you have RNA, you have proteins, and you have metabolism, and then you have a flow of information all the way from DNA to proteins and eventually to metabolism. Well, in synthetic biology, I mean, it's good that you keep that in mind, but this is not very useful. What is really useful in synthetic biology is this part here, is that parts, biological parts, once they are composed, they form devices. Once the devices form, they can be assembled to form modules, and eventually modules can form systems, okay? So this is a different perspective of the same thing. Again, this is something that I showed you on passing the other day, and is what are the different modules that are involved in formation or in assembly of symbiote devices. And then basically you have to consider all these parts, all these components. You need a promoter that receives an input, and then it releases an output that goes into RNA, and you need a part of the subsequent DNA that is not transcribed, it's transcribed, but it's not translated. This untranslated leading 5'. Then you have the gene of interest, and you have a 3' region that is not translated either. So each of these blocks here are subject to a degree of standardization. And in practical terms, what you have, and this is a bit abstraction, then this is a lesser abstraction, still an abstraction, because you have this biosensor module that has the regulator that receives a signal, in many cases an inducer, and then it gives out pops, and then the pops enter into an operon, one or more genes, and then at the end you have a pathway. That is what you want, and at the end you have to have a terminator. So this is the kind of super typical, super basic synthetic biology device. Now, where do we put the devices to have them working in a given biological system? Well, basically you have two choices. One of them is assembling the devices in a plasmid, and we will discuss later how that works and what are the pluses and minuses. But also, and I am anticipating things that I will tell you later, we can also put the devices, the artificial devices, the engineered devices, in a transposon. So transposons are mobile genetic elements that go into the genome of the target bacterium or the target biological system. And then the advantage is that the DNA that we engineer in the laboratory, instead of being expressed and located as an extrachromosomal element, goes to form part of the genome of the chromosome. Okay, I will return to that a bit later and give you an aperitif of things that I will discuss subsequently. Now, in synthetic biology, we have a problem, okay? And it's not a trivial problem. And it's the problem of context dependency. So in what you may call engineering, in traditional engineering, the parameters and the transfer functions, input, output of every device is fixed. So in most cases, in general, the activity, the measurements, the parameters delivered by a given device is completely context independent. If I have a resistance of 5 ohms, the 5 ohms will be 5 ohms here, there, in this transistor, in this radio, in this other refrigerator, in this other washing machine, it will be always 5 ohms. However, in biology, it is something that we have to deal with. The values associated to the different devices change with the context. So there's this issue of context dependency. Unless what we enter into the system is completely orthogonal, and therefore the activity will be completely, or to some extent, independent of the context. So which are the circumstances that alter, what are the contexts contexts that change the parameters associated to a given engineered device? Well, you have a bunch of them here. You have different layers. Well, first of all, you have a short range text. You may have a device, depending on where it's in the chromosome, depending whether it's in a plasmid, depending where it is in the general, say, genomic contents of a cell, the behavior can be different. But also, there's echo here. Well, so, but also it depends on the number of copies that you have that device. It's not the same to have something in the chromosome as having it in a plasmid that normally is multi-copy. It depends on the genomic location, also in the cell location. And one of my last talks will be exactly about this issue of the cell location. So you may believe, or you may think that cells, bacterial cells, are completely homogeneous. Well, they are not. They are not compartmentalized, but they do have domains that are separated by means of other interesting forces. And then also you have an issue with growth condition. It's not the same when you grow cells fast, when you grow them in minimal medium, when you grow them in so there's an issue with that. Then the host species, you may have the same device expressed in E. coli, expressed in Pseudomonas, expressed in Salmonella, or expressed in a plant, and then the host will make a big difference. And also the physicochemical settings. So are you in a place with a lot of humidity, with higher temperature, with lower temperature? So you may say, well, then this creates an impossible situation because you are very happy making your device here, but then how it will behave going through all these layers might change everything. Well, this is one of the challenges, and as you will see later, it in principle can be managed. In some cases, can be managed. In other cases, things are more complicated. Okay, so let's take a look to what would be the ideal, completely ideal situation. So let's suppose that we are very competent synthetic biologists and that we want to develop a pathway that will convert this product into this, or this substrate into this product. For instance, you may have, an aromatic compound from petroleum. You want to produce a super expensive drug for cancer therapy, for instance. Okay, so if you were an engineer, I say, well, this is a piece of cake. You take a regulator that can be induced by an inducer of my choice, IPTG, or by heat, or benzoate, or whatever. And then I engineer a promoter in front of a series of genes in which each of the genes deliver an activity that is important for the production of this said product. And then as I have many genes, and then each of the genes may have a different model, a different origin, then I engineer these intergenic regions that in many cases will be ribosomal binding sites or Schindler-Gardner sequences. Remember that we mentioned that when we talk about the gene expression flow. Okay, so this would be the ideal situation, right? I say, well, you say, well, to go from this to here, I go to the literature and I need all these enzymes. I go to the genes, I clone them, I put them in front of a strong promoter, and bingo, it should work. Okay, well, this is in many cases what we kind of tell the world, but ladies and gentlemen, things are not so easy, right? So in many cases, we think that we are doing fantastic, but we are not doing fantastic, okay? We have to be careful about that. Okay, why we are not doing very well? Well, mostly because of these 4 reasons. Number 1, biological components are context dependent. I just mentioned that. Also, and don't forget that we are talking about biological systems that are alive, and therefore they are subject to Darwinian evolution, okay? So that means that they can mutate, and the mutants in some cases can overcome or can be more successful than the, say, constructs that we make on purpose. But also, everything that we construct in living system interacts constantly with metabolites and chemicals. So if we produce a protein, the protein does not appear out of the blue. It's all surrounded by a big, say, concentration of other molecules, other chemicals, and other things. And what is more worrying is that in biology, combination originates emergence. So it's not just that we have context dependency, is that that may vary a little bit the input-output functions. is that sometimes when we combine biological components, then the outcome may be completely unexpected. So it's not that one thing is a little more, a little less. So this is a serious problem, fascinating problem that goes as follows. In mathematics, 2 and 2 is 4, okay? No discussion about that. In engineering, things are not so, because 2 and 2, this is very difficult. When you, for instance, make a screw turn, you have a dimension of the receiving part exactly the same to the one of the other. And then if you had exactly the same dimension, it would not work. I mean, you need to have some little space between the two to make them fit. This is something I will return in a moment. It's connected to the allowance of standards, but you know, in engineering, things are close to mathematics, but they're not mathematics. But in biology, we have this thing that is 2 plus 2 may be 4, but then you have this phenomenon that is emergence. Emergence is that you don't know what's going to happen. You cannot predict what's going to happen when you put these 2 plus 2 together. So sometimes you combine one gene and other gene, and you think that something is going to happen, and then what happens is something completely different. You put this enzyme and this other enzyme, and you believe or you predict that the result of these 2 steps will be one specific reaction, and then what happens is that maybe the first enzyme will kill the other or will inactivate the other. In other cases, it will activate the reaction, and in other cases will originate a completely novel reaction that has nothing to do with the ingredients. So this is emergence, okay? So there's this famous quotation from Drew Endy. This name I have been mentioning various times in this course because he's one of the founding fathers of synthetic biology. Say, engineers hate complexity. I hate emergent properties. I like simplicity. I don't want the plane I take tomorrow to have some emergent properties while it's flying, okay? And I think it's a very good quotation. So in biology, we have to live with this emergence of new properties, and we may discuss whether they should be welcome or they should be suppressed, okay? But the thing is that this is a main difference between biological systems and engineered systems. Okay, let me spend a few slides on the problem of how to fit 2 components that are standardized, okay? So as I mentioned before, if you, I mean, having a dimension like in mathematics that is completely precise is impossible. You always have to play with errors. So in mechanical engineer, in compositional standards, there are 2 important concepts. One of them is the concept of tolerance, and then is the concept of allowance, okay? So tolerance is the margin of error in the dimension of an object or property in respect to a nominal value that is connected to the inaccuracy of the production process. So I want to have this piece 2 centimeters, but it will not be 2 centimeters. It will be a little more of 2 centimeters or a little more or a little less of those centimeters. And we have to live with that because we are not in a mathematical work, we are in a physical work. But then we have this other aspect that is called allowance. So allowance is a deliberate flexibility that you enter in the dimension of an object to make sure that the 2 things fit together, okay? So when you have a screw, component, and you have the receiving hole of the screw, then, I mean, if the 2 were absolutely identical, the system would not work. You have to leave a little room for the 2 things to go after the other, okay? So this is something that the engineers know for a long time. And the thing is that, for instance, if a shaft had to be inserted in a machine and 10 millimeters is the nominal dimension, number one, there will be some error, and therefore the system has to be, say, thought to tolerate the kind of error margins that are associated to the production process. And then, by the same token, and I don't want to go in detail through this text, is that in many cases, to make the system work, then you have to allow some margin of, not of error, of room for the 2 things fitting together, okay? So remember, then tolerance is tolerance to errors, and allowance is doing something deliberately that the parts a bit for from perfection just for the sake of securing that the standards enable putting two things together without a problem. And engineers have developed this type of tables for many years. So what is the kind of deviation that you have in one of these tolerance, parameters versus the other one's parameters? And this is something that engineers use all the time. Okay, this is for mechanical engineering, for electric engineering, and for kind of the engineering that we know on a daily basis. How can we translate this to the biological world? Okay, because obviously, if we're talking about standards, we have to consider all these circumstances. Okay, so let's recover again this metaphor of the radio built with parts and see how far we can project these ideas that we have just mentioned about engineering standards to the biological world. This slide that I also showed the first day indicates that the radio has many components, many parts, and also cells from the perspective of synthetic biology can be understood as a bunch of structure and well-organized and well-characterized parts, okay? So in this case, the parts are transistors, cables, connections, bubbles, and then you have this speaker. Then in the case of a cell, you have genes, you have, I'm sorry that the resolution is not good, but you have different types of molecules, you have controlling elements, you have a list of them, and the list is very long, but it's still limited, it's not infinite, so it has a number of things. Okay, so we return to kind of the star concept in synthetic biology, that is the concept of parts. So what is a part? A part is a DNA sequence that encodes one non-equivocal biological function. It has to be one. It cannot be 20. It has to be one. But from the perspective of synthetic biology, this is not enough. So the part has to be endowed of additional properties. Number one, the part has to be composable. That means that the part has to be thought to make it amenable to further composition of this part with other parts, with other segments of DNA. It has to be reusable. Reusable means that the part is good not for one specific purpose, but one day I use it for making this construct, then I take the part some other day to make it another, and another, and another. I can reuse the same part for various purposes. If I have a promoter, then I can use the part for that promoter part for expressing, I don't know, beta-lactamase, but tomorrow I want to express a pathway for degradation of silane, and then I can use the same promoter, and the other day I want to do it for production of genes or expression of genes that deliver at the end some synthetic material. Okay, so it's the same part, and we use it in different contexts. Then it's scalable. Scalable means that you can use it in many different places. It has to be standardized, and I will return to that. And also, and this is important, it has to be open access, okay? So, well, this is not obligatory. I wish it were obligatory, but it's not obligatory. But in general, the community of synthetic biologists advocate, including myself, that biological parts that are standardized should be made available to everyone without the limitations of intellectual property, patents, and all the rest of it. So what is in a part, okay? Okay, in a part, you have basically three things, three say, contents. First, the DNA sequence, of course. If you don't have a DNA sequence, you don't have a part. Then the function associated to the biological, to the DNA sequence, and also the activity figure. And the activity figure is a very important, say, characteristic of a biological part, because if not, you are not in synthetic biology, you are before synthetic biology. So you need to have a sequence, a function, an activity figure. For instance, I have this sequence of DNA. There is a promoter. The biological function is a promoter, and the activity of this promoter is 40 POPs. That's a super wonderful part. It's super well characterized, okay? So I'll show you how these parts can be abstracted and how far you can get with this type of definitions. So, well, one part, for instance, and also, as is the case with electronic components, you can give them a code. So you have a list of parts, that are encoded, not with the whole sequence, but just with a name that again is standardized and enables you to handle them very easily. So this would be the perfectly defined part. Okay, so you have a sequence of DNA. The function is a promoter. The family belongs to is the protein DNA interacting proteins. The activity is 2 POPs. Then requires this other part to be assembled. Probably this part is a plasmid or something, I don't know. Cell type, any, that means that it can go in any say, biological system. Temperature is appear to say that this go up kind of lower than room temperature. Issues known, in principle, no one has found any problem with it. And then public, that means that everyone can use it, okay? So this is just one part. There are thousands and tens of thousands of parts that are characterized under this kind of nomenclature. But then remember this idea that the parts have to be composable. So composable means that a part of the part proper, you have to flank the part with DNA sequences that enable an easy composition of this part with another part. Well, as you may recall, these days there are many different ways of assembling DNA, and in this case shown here, this is through restriction ligation process. Okay, this will be a restriction-based assembly of parts. Now there are many other processes, but in any case, a part of the part has to be decorated at the sides with some sequences that enable the composition of this part with other parts themselves. So, for instance, and the composition has to be such that when you do it, you generate another, say, composed DNA that itself can be recurrently associated or added with other parts. Okay, so you have here, for instance, part 24 and part 25. You digest with a specific restriction enzymes here and there, and then what happens is that this part will go here, but at the same time it will reproduce the enzymes that you had at the beginning, and therefore you still have some parts at the side that reproduce what you had at the beginning. However, the part that you had here to make this linking between part 1 and part 24 and part 22 is engineered in such a way that once the DNA gets mixed, then there are no restriction sites any longer here. And therefore now this behaves as a single unit that can be subject of subsequent rounds of addition of other parts, okay? So this is one of the many ways of adopting standards for DNA assembly. But remember, and this is also a slide that comes from the first days, that parts is not the end of the story. Once you have parts that can be assembled, you can produce devices, and devices can be assembled to produce systems. You may remember, I'll show you a couple of slides that I shared with you the first day, that for instance, in this case, you have a device that is an inverter, because we have a lac repressor repressing expression of another repressor. You can define very clearly this function, and you can abstract this construct in the form of an inverter. Remember the first day that we discussed the repressilator that is based in 3 inverters, and so this is something that is once you have parts and the parts can be composed, then you can start composing them to produce devices and so on. But there's this part that we did not discuss the other day, and is the characterization. Okay, so every part, for instance, you have here a promoter that responds to a molecule of current sensing. That is a part. It has a code. It has been engineered upstream and downstream with some say sequences for further composition. But how do we characterize that? Well, there are various initiatives, and there have been indeed, mostly in the US, this idea of collecting thousands and thousands of parts and characterize them in terms of inputs and outputs. I mean, this obviously for a human is impossible. It will be very boring to spend all your time making this type of characterization. But many of these operations to define the input and output values of a given device now can be run by a robot, in such a way that at the end, for every part, and there are thousands of parts, you can have a kind of information page where you can really monitor and visualize all the characteristics and all the say parameters associated to that part. And then with these characteristics and these parameters in hand, then you can go on and make devices and the rest of it. Okay, so this is connected with the what I have told you so far is connected with the standardization of parts. But there are other, say, aspects of synthetic biology that also deserve and have been standardized. One of them is the language. And the main language for describing synthetic biology constructs is the so-called SBO. SBO means Synthetic Biology Open Language, and is a language that has like different degrees of, say, depth, and is very useful because it enables you to describe genetic constructs with a language that can be understood not only by humans, but also by machines, by computers. So that's something that is very useful. And the SBO platform is becoming, or the SBO language is becoming more and more used by the community to have an accurate description of different procedures and different parameters associated to a given biological construct. So in such a way that instead of having this soft definition, this soft wording of some biological phenomena and some biological properties, then with SBO, we have a very precise description of what is going on. Not only that, but there are some initiatives in the last few years to convert the SBO language in a language that can be fed to a robot to run experiments itself. So instead of having to do the experiments ourselves, what we do is that we code the experiment in this language, and then a robot can really run the operation without any human intervention. So the most superficial, say standard associated to SBO has to do with the annotations, the visual annotations of the different biological activities that are at stake in a genetic construct. Okay, so I don't know whether in your biology classes you represent promoters or genes or whatever in a kind of non-standard way. But if you want to follow the rules, then the community has decided that you have to show your constructs following these visual standards. So you have a promoter, you have to use this little arrow that goes in a straight angle and in perpendicular to another little rod there. Okay. You have a gene, you have to use this annotation. You have a ribosome binding site, is this annotation, terminator, operator, insulator, blah, blah, blah. So that means that when you say, well, I have a gene, I have a pathway, whatever, you cannot use, or you should not use any representation that you fancy. No, please use always standard annotations for graphical representation of your constructs, because that enables you to communicate the contents of your constructs to a much wider community that will not be confused by using some whimsy, symbols there. Okay, so the next say screw turn is that we have defined the parts. We see that the parts have to be standardized, that there are some kind of characteristics and so on. But then where do we find the parts? Okay, so basically, well, parts can be made by any laboratory and you can have them, etc. But there is a major repository of parts that is found in association to IN. I will go to that in a moment. But there are catalogs in which you can find different categories of parts standardized in a way that enable their assembly and so on. And the most famous of all of them is this registry of standard biological parts. And you can go into to find out what they are and what they have in this page here. At the end, I have a number of web pages, links that you can check. And well, it's constantly updated. And what you have there is an immense collection in the order of thousands, tens of thousands of different parts that the community has developed over the years following these rules about having a DNA flanked with other enzymes that enable their composition and so on. And that's something that is very, very useful. So for instance, what type of parts you have there? And you have many, many different, say, categories. Well, you have some collections, for instance, of say, promoters, more or less used parts, reporter proteins, quantified promoters. I mean, you have a whole bunch of them, and this is just a screenshot of a web page, and if you go one by one, you will have exactly all these things. And also, what is interesting is that in this registry, you can get what are the rules for producing these parts. So that means that if you, in your laboratory, develop another part and you want the part to be considered a synthetic biology part, then you have to follow these rules and eventually add that part to the collection so that everyone now can start using it for the different purposes. Okay, I have to say in this respect that not all parts are equally good because they are not curated. Not curated means that you say, well, here you produce in your laboratory around the corner this promoter, and then it works one day in your hands, and then you send it there. And well, it may not be properly characterized and so, but still they will receive a code. However, it's not completely sure, it's not well documented that that promoter will work as expected. So that means that you have a whole collection of different parts. They are being used by many different people. And at the end, some of the parts are used by different communities, and then they are very well documented. So out of the thousands and thousands of parts, there may be around 200 parts that are really, really well documented. The rest might be junk in many cases. You have a lot of noise in the parts that you deliver to the collection, but in some cases, some of them are really, really well documented, and they work very well because different communities have used them, and in principle, you can trust them. So, well, there are some, if you go here, well, this is really kind of funny exercise. If you go to this collection, you can see what are the best documented parts, which are the best demanded part, what are the best plant chassis, what is, I mean, but it's funny if you go to the well-documented parts or the frequently used part, you see in reality which are the ones that people like to use much better because they are much better characterized. And some of the most demanded parts are promoters. So these are the typical part that everyone would like to use for one particular application. And interestingly, the promoter that is best demanded is like top one, top one promoter in town is the PBAT promoter. That is an E. coli promoter that is regulated by arabinose, okay? And it's very, very well regulated. So that means that in the absence of induction, the expression level is very, very low. It is not zero, but it's very low. And in the presence of arabinose, that is the inducer, you have a high level of expression. But you have other promoters here that are kind of curious. For instance, there's this promoter that is a constitutive promoter. So that means there is a promoter that will deliver POPs without any, say, other, type of regulation. You have other promoters that respond, for instance, to quorum sensing. Remember, we discussed quorum sensing. You have other constitutive promoters. You have the promoter like Y. In many cases, if you don't know much about bacteriology and microbiology, many users like the lag promoter, because the lag promoter can be easily induced with IPTG, an inducer, but in fact it's not very good. So it's not, the regulation is not fantastic. Well, you have these very well documented promoters and so on, but there are many other parts that are, are used and documented to different degrees. So once you have these parts, and then you have different vectors, then you can assemble them, these bio bricks that I told you yesterday, or I anticipated to talk about them a few days ago. And then, well, you start with a plasmid, and then you follow the rules. You can use the same vector plasmid to keep on adding parts and parts and parts to a given construct. And this is something that it was a conceptual revolution when it was proposed, because before this, it was not considered that DNA assembly could be subject to any degree of standardization. Well, if you go through a web page, they have some rules. I will not go through the details. And well, it's just to that you follow some rules, not just a a kind of crappy, no, that your kind of whimmy say desires, but you just use rules that you take the plasmid, you carry it with here and there, and then the next one has to enter through this enzyme and so on and so on. So that means that you follow a kind of roadmap, a kind of say a pipeline for production of constructs that is entirely standardized. And this idea of standardization is the very basis of IEM. And you may recall that the other day I told you a bit about IEM. So IEM started time ago as a competition, and now it has grown at very, very large dimension. And now it's over 10 years, the kind of IEM competition and so on. And if you are, I told you the other day, I will not tell you again. So if you want to know more, just go into the IEM webpage and you will see the kind of range of different projects that you have in this type of things. Every year, as I also said the other day, the community of undergraduate students that register or enroll in IEM get together, form in a jamboree that at the end becomes a kind of a strong competition. So it's why some people say it's a synthetic biology battlefield. You have many teams from all over the world that get together, and at the end there are winners and so on. So this is this lady here worked in my laboratory at some point, but it's a very good training exercise. And remember that for the last part of this course, I will ask you to go through some of the projects and get inspiration, and ask you to propose, not with all the detail, of course, but some type of device, some type of genetically engineered machine or life system that you think could be good to consider for future endeavors in the field of synthetic biology. At the end, people get certificates and people get very happy and all the rest of it. Okay, so that would be the kind of general story about standardization. Now what I'm going to do, before we make a real break, is just to advance what you may call the low-hanging fruits in standardization. What things can be standardized very easily, or at least without great complications, because other biological, items can be difficult to standardize, but others are relatively easy to standardize. And the easy things that you can standardize is, to start with, is the DNA segments that bear specific activities. So the function-bearing implants, this is something that can be standardized, and BioBricks is one of the cases of standardization. The other thing that can be standardized, and that will spend for the rest of the talk today, is genetic tools. Genetic tools can be standardized very well, and you will see the advantages of standardization of genetic tools. And then what you can standardize also, and we had a big discussion on that in previous days, is the destination chassis, okay? Whether the chassis at the end or the recipient is a natural bacterium or is a bacterium that has been reshaped, the genome has been remodeled or rewritten or modified somehow to receive a specific vectors that on turn contain a specific fragments of DNA that have been engineered for given purposes, okay? So that's how things would go in the case of the Biochemical standard. And I leave you with this slide that we will discuss in detail in the second part of the class today. Okay. Now we have a 5 minute little break, and then we go back into this story about the SEBA platform. Okay.